Hi guys, I'm Adiba and I was just going over the topics from Cell Bio. This is for the first exam. In this one, we'll be going over the structure of DNA, chromatin, and chromosomes. Just the DNA structure itself, you're starting with the 5' and ending with the 3'. You have two phosphate groups and they're usually on the third carbon. And on the third carbon of the other one, there's another phosphate group. So essentially, just remember that there's a phosphate group on the third carbon. On the fifth carbon is where the base is attached, and then the second is where you have the CH2 attached. So main point of importance is that the third carbon has the phosphate group attached to it. On the fifth one is where the base is attached. You have the sugar itself. So this is just the DNA double helix. You have at GC, and between them is the hydrogen bonds. You have to know that at there's 3 bonds, and between GC there's only 2 bonds. Actually, I scratched that. It's the opposite. Between GC there's actually 3 bonds, and between AT there's actually 2 bonds. One way I would remember this is because AT comes first and then GC. This is how the acronym goes, at GC. So at, because it's starting first, would have the smaller number, so 2, and then GC would have 3 hydrogen bonds. So for add, there's just 1 hydrogen bond between NH, 1 hydrogen bond between OH, and for GC there's 2 OH bonds and then 1 NH. And this is just another diagram for that. Moving on, this is just showing how DNA double helix and have different genes, and then you have through transcription you get RNA, and then it turns into protein. So only about less than 3% of the coding RNAs actually turn into protein, whereas on the other hand, all the other ones are non-coding proteins. Essentially, they don't really do transcription or translation per se. Some include like less than 200. It's like small SN RNAs, and then you have SN me something, and then SN and SNO RNAs. And then if you look at the greater than 200 mark, you have large SN RNAs, long ncRNAs, my bad, which has link RNAs, eRNAs, GSRNAs, pRNAs, etc. So this is something I want to ask if we have to remember, right? So now we're going over genomes and chromosomes. There's 46 chromosomes in the human genomes, which can individually be labeled and identified via fluorescent microscopy, right? And then very similar genomes can evolve different chromosomal number and organization. This goes back to the fact that, again, there can be multiple genes in one DNA, and they can give different results. Chromosomes are like balls of yarn with varying degrees of organization throughout the cell cycle. So you have the interface, metaphase, interface again. So S1, M, and then S2, right? So you have here an interface chromosome. You have the nuclear envelope surrounding the nucleus, and then you have the gene expression and chromosome duplication. So that's been duplicated. And then interface, you can see more in depth here, a lot of colors. And then in metaphase is where there's the mitotic chromosome, there's spindle, and then cell division happens, and then they actually multiply. Nucleosomes, one thing to remember is that hold on, I'm just gonna confirm, like the S phase is where the duplication happens, right? Sometimes you question like the basics. Actually, I can just do it here. Yeah, so the S phase is where the duplication happens. You have the G1 phase, cell growth, and then S phase is DNA is duplicated. G2 is where it grows more, and then M is where they separate it into two different things. So S is the only place they would actually. S phase is the only phase where they would actually duplicate. BMAT, prophase, anaphase, metaphase, telophase. Interface, where is interface coming from? Nucleosomes are formed by histone proteins and DNA. So you have H2A, H2B here, H3, H4, that's making a histone octamer, and then that's being broken down. So these are the two histone complexes, and then you have the DNA double helix. As you can see, there's a lot of different colors of histones. They're all different types. They're all making one complex. So essentially, nucleosomes are made of histones and histone proteins and DNA. Chromosomes are made of DNA filaments packed into chromatin fibers. Here you have a 2 nanometer, you have 11 nanometer. These are beads on a string to form a chromatin. So these make a chromatin. And that chromosomes are made of DNA filaments packed into chromatin fibers. DNA filaments. When you take DNA filaments, so you're essentially making a lot of DNA filaments come together, and that is what is giving you a chromatin. Chromatin fiber, and then chromatin fibers are formed into loops, and that is giving you a metaphase chromosome. Chromosomes made from DNA filaments. Chromatin fibers. Chromosomes are made of DNA filaments packed into chromatin fibers. DNA filaments packed into chromatin fibers. So you see that you have really narrow, like small chromatin here, right? Chromatin, and those are coming together. They're making these loops, and that is what is giving you a metaphase chromosome with the whole centromere. Nucleosomes are big enough that they can be seen under electronic microscope. So you have the double-stranded DNA helix, the N-terminal of the histone fall, then you have all these different histones, right? And then histone proteins, nucleosomes, and chromatin and chromosomes. So nucleosomes can be seen under ES, electron microscopes, right? So a condensed chromatin fiber, and then you have beads on a string chromatin. So essentially what they're saying is chromosomes are made, well, okay, first of all, DNA is made from histone proteins. No, nucleosomes are made from just histone proteins and DNA, right? That's what we came to conclusion. And then chromosomes are made from DNA fragments. Not DNA fragments, just DNA condensed together, DNA filaments packed into a chromatin. So you see that here, where there's a condensed chromatin fiber, where there you have all these DNA filaments making a chromatin, and then the chromatins being packed together, and then you have the chromatin fiber, and then you have the beads on a string chromatin, which is much thinner and is just essentially before all that integration happened, and you got the condensed chromatin fiber. Euchromatin, heterochromatin, and nuclei. Now this is important because here we go over how if there is sorry, scratch that. Let me just go over this. Nucleoli is the site within the nucleus where actively transcribed regions of the chromosomes converge. So you have the actively transcribed region and they converge in this site. I just repeated what I said. They're highly rich in RNA and proteins as opposed to regions containing heterochromatin, mostly transcriptionally silent. And euchromatin regions are less actively transcribed. So you have this heterochromatin region, which is dense, right? Dense, but it is transcriptionally silent as compared to actively transcribed regions where there's RNA and proteins. Heterochromatin is dense, it's found in the centromeres and telomeres, it's transcriptionally silent. Euchromatin is actively transcribed but less than the nucleoli, and then you have the nuclear envelope, the nucleolus, chromosomes, nucleolar RNAs and protein. Sorry about that. So heterochromatin is dense, it's found in the centromeres and telomeres, and it's transcriptionally silent, whereas euchromatin is actively transcribed, right? It's an active site. However, it is less in the nucleoli as compared to heterochromatin. Then you have the nuclear envelope, the nucleolus, the nuclear RNAs and proteins, the chromosomes. As you can see here, it starts with the heterochromatin, right, with the telomeres and stuff, telomere and the centromere, right? And then you have the euchromatin, which is actively transcribed, and then you have the heterochromatin again, which is centromere, euchromatin, heterochromatin, euchromatin, hetero. So it starts with hetero, then goes U. One way I would remember this is HE, he. So H for heterochromatin, which is transcriptionally silent, and it's mostly found in the telomeres, which is at the ends, right? And then the middles, the centromeres. And then you have euchromatin, which is actively transcribed, but it's less in the nucleoli. We're almost halfway through. Actually, more than halfway through. Histone modifications and chromatin remodeling. You have the N-monomethyl lysine here with the methyl group, then N-methyl arginine, and N-dimethyl arginine, it's asymmetric, and N-dimethyl arginine, which is symmetric. Now all these details, N-acetyllysine, O-phosphoserine, and N-dimethyl lysine, and an N-trimethyl lysine, and these are all, I believe, are helping forming our histone here, right? All these different little tails. This whole thing is just H3 histone. Histone modification, chromatin remodeling is what we're talking about here. So I will jot this slide down, which is slide 14. Go over again. Mechanisms of chromatin remodeling. Hopefully this will clarify slide